Cellular Genomics and Bioplatforms Australia. He has also acted as an advisor to the New South Wales state government and the government of New Zealand. He has been elected to the Council of HUPO, the Human Proteome Organization, 4 times. He's a current council member and was co-organizer and program chair of the International Congress HUPO 2010, Sydney, and a co-organizer of HUPO 2019 in Adelaide. He's an elected member of the Management Committee of the Australian Proteomics Society and a co-organizer of its annual conference. Professor Marc was a senior editor of the Really Journal Proteomics for a 10-year period. He also played an important role in industry. Professor Marc has co-founded 2 biotechnology companies, Proteome System, which is established in 1999, and developed proteomics technology and platform for rapid point of care diagnostic testing. And another company, Regenus Pty Limited, established in 2007, has developed a direct stem cell product which has recently been licensed to a major partner in Japan. Professor Marc has been the director of the Ramakirti Center for Genomics since 2011, a center which is now the largest at any Australian university. Their long background and more. So without further going, I would like to invite Professor Marc to share with us his work or his talk today with the title, The Intracellular Network of Protein Methylations, Enzymes, Motifs, Crosstalk, Regulation, and Function. And we look forward to learn about the methyls transferase or protein methylation in this webinar. Welcome, Professor Marc. So the
Thank you.
WebEx is yours.
Okay, thank you. Can you hear me?
Yes, I can hear you.
Yes. Now, unfortunately, we may have that same technical issue again. I don't have the share screen in the bottom of this WebEx. So, if it's possible to make that change that we made a minute ago to the other room, that would be fantastic. Oh, here it is. Okay, good. We've got it. We've got it.
You got it, yeah?
Yep, wonderful. Okay, so
great.
Okay,
Yes.
so can you see my screen okay?
Yes, the screen is clear. And I can see your mouse, your laser pointer.
Good. Okay, so I think we can begin. So look, thank you very much for the invitation and the chance to tell you a little bit about the work that we're doing here at the University of New South Wales in Sydney. And you know, it really is a great honor to be able to talk with you again. I think last time that I had the chance to talk with you is almost 8 years ago now. So thank you so much for the invitation, and I'm really pleased to be able to catch up with some of you and talk with old friends. It really is a great privilege. So today I'll talk about protein methylation. And it is something which we've studied in a lot of detail for it's almost 10 years now. And in particular, we've had some objectives in terms of our work. We are systems biologists, and we are interested in how networks are put together inside the cell. We're interested in how networks function and their regulation. And so what you'll see in this presentation is how we've built a network over a 10-year period of time, and then sought to understand its function and its regulation. So that's I guess a broad picture as to what we're going to talk about in this presentation. So some of you will be familiar with methylation, others won't be familiar with methylation. So I'll give you a little bit of a background into this quite interesting and unique post-translational modification. So the methylation of lysine is a very common modification inside the eukaryotic cell. It does exist in three forms. You can see the mono, the di, and the tri form of the modification. And inside the human cell, there's at least 150 methylation sites. It's not a very large number, but it's not a small number. Now, methylation itself is actually, it's called an expensive modification that the cell needs to spend 12 ATPs per event. So it's a much more expensive modification than phosphorylation that involves just one ATP. Now, the methyltransferases, there's a methyl donor called adomet inside the cell, and there's demethylases that can remove the modification. So it's a reversible modification, and it can act as a switch in terms of it being on or being off, as well as being in these different forms. So it's a really interesting modification because sometimes these different forms have different functions. Now, there are lysine methyltransferases, but they're interesting in that they actually have one or few substrate proteins, and that's different to what we see for some of the other types of methylation, like arginine methylation, that we'll speak about in a second. And when you add methyl groups onto lysine residues, what you do is you actually maintain the charge. You can see how the charge is maintained of these amino acids, but the side chain of the amino acid actually becomes more hydrophobic. So it's quite important in terms of changing the type of the amino acid that you've got. Now, what we know is that, as I said, about 150 proteins inside the mammalian cell have lysine methylation. But of course, the histones are the best characterized form of protein that have methylation. And you can see there's methylation on histone 3, there's methylation on histone 4. On histone 3, there's 4 sites, and you can see them just as I'm illustrating here. And all I'm really showing you here is that methylation, it does tend to be actually found a lot on proteins of related function. Okay, and we'll talk about that a little bit as we just get further into the talk. So when you find proteins methylated, chances are there are related proteins that are also methylated. It's really a feature of methylation and how it works. Now, as I mentioned, the human cell, the human methyltransferases, there's a lot of methyltransferases of lysines inside the human cell. There's 58 histone modifying enzymes, for example, alone. So you can see it's quite a complicated modification, and you can have in the human cell a lot of methyltransferases that act on the same histone. So from a systems perspective, this is quite interesting. You have many enzymes that can modify the same site on a histone, and this is quite unique in terms of how this system actually works. So many enzymes coming into one particular modification type on the histone proteins. And you'll see how that's quite different to how the arginine methylation events work in just a few slides. Now, in terms of the role of lysine methylation, once again, just background for you, there's a number of processes that lysine methylation is found in, but there is one common theme in terms of what it does and how it actually functions in a physicochemical sense. In that all of these things that we see here involve the modulation of protein-protein interactions. And that's a really important thing for you to remember in terms of its role. And it's really important, once again, to think about methylation as being reversible, because now you can see that this is potentially a switch to control protein-protein interactions. And you'll see that that's very important as we get further into our talk. Now, methylation of arginine, this also exists in three forms, a monoform, a symmetric and an asymmetric diform. It's actually more widespread than methylation of lysine. About 2% of arginines, for example, are methylated in the nucleus of the rat. There's more than 1,000 sites known to be present inside the human cell. And whilst you typically have one lysine methyltransferase that can act on one site on one substrate, arginine methyltransferases can have many substrate proteins. And so there's only actually 9 arginine methyltransferases known inside the human cell, as compared to the very large number of lysine methyltransferases that are known to exist inside the human cell. Now, once again, the charge of these amino acids is maintained, but you change the side chain of the amino acid. It becomes more hydrophobic, and particularly it shows a capacity to stack and to interact with tryptophan. And that's very important in terms of how this modification is actually recognized by certain domains on other interacting proteins. Now, in terms of the role of this modification, once again, this is just important background for you. What you'll see here is largely with epigenetics, with the messenger RNA splicing, the DNA repair, proteins that are involved in the and are actually methylated on their arginines are involved in interacting with and modulating processes associated with nucleic acids, with either DNA or with RNA, or with proteins themselves that are involved in the interaction with DNA. Now, whilst many of these roles are missing in lower eukaryotes, certainly the messenger RNA processing and transport that is highlighted here is conserved throughout eukaryotes, and arginine methylation has a really important role to play in that process. And we'll talk a little bit more about that as we get into this presentation. So that's some background for you in terms of what protein methylation is. And I suppose what I wanted to then pose is really these 4 or 5 questions here, which I'll talk through and talk about our research in these areas. Now I've given you this view on what methylation is and some of its role inside the cell. So certainly about 10 years ago when we started this research, it's about 11 or 12 years ago now, um, we were quite frustrated in that we wanted to look at this as a system, but we needed to, of course, find all of the sites of methylation inside a eukaryotic cell. That was one objective that we had. Then what we wanted to find was the methyltransferases that are responsible for those precise sites of protein methylation on the proteins. And so if we could do that, then we were asking a question, can we then build a complete network of protein methylation for a eukaryotic cell? So it's kind of like a signaling network with phosphorylation, but involving methylation and methyltransferases. And these 2 points are the things that I'll talk about first in this presentation. Okay, so to actually make this happen, to build a network, what you need to do is to be able to map your methyltransferase enzyme to a substrate protein. And there's 2 things that we needed to do to make this happen, and then to kind of do it at scale so we could build this complete network. Certainly, we wanted to be able to look at genomes and to be able to predict what the methyltransferases actually are in those genomes. And luckily, I mean, there is a common methyl donor. It's called ADOMET inside the eukaryotic cell. And so you can look for ADOMET binding proteins inside the eukaryotic cell. And there's actually 3 main domain types that are involved in that. One is called set domain. There's a 7 beta strand and then a spout domain. And you can predict the number of these proteins that exist in, for example, the human genome, Homo sapiens, or in the yeast genome, Saccharomyces cerevisiae. But whilst you can predict that proteins are methyltransferases, it's actually very difficult to know what type of methyltransferase they are, whether it's a methyltransferase that acts on lipids or acts on nucleic acids or acts on proteins. And so what you then need to do is to be able to use some type of technique to then modulate these different proteins or genes and find their effect on methylation sites. So at the same time, we need to be able to do methylation site discovery, and this is where the proteomics work that we do comes in. And there's a range of tools that you can use for this, and I'll talk to these in some detail. But I guess I did just want to mention there's 3 or 4 of these: proteome arrays, false discovery rate control, something that we call methylquant, which we developed inside my lab to help us with methylation site discovery. And ultimately, we then used these, and I'll talk about these experiments, where we've done a whole series of knockouts of either yeast genes or of some human genes using CRISPR-Cas9 approaches. And when we've knocked out the genes of methyltransferases inside the cell, we've then been able to look at the proteome to find loss of methylation sites. And from that to map our methyltransferases to methylation sites inside the yeast cell or the human cell to a lesser degree. And after these loss of methylation studies, we've then done largely gain of methylation assays where we've done in vitro analyses. We express recombinantly the methyltransferase, express recombinantly the target protein, and then look for gain of methylation using mass spectrometric analyses. Once again, another couple of new approaches that I'll talk about in terms of how we do this type of work. Okay, so just to summarize that once again, a lot of the work that we've done to find methyltransferases and proteins that are methylated associated with that involves the knockouts of methyltransferase, and we've largely done this work in yeast because of the excellent genetics that yeast has, and it's got a reduced set of methyltransferases and methylated proteins. And of course, you can generate knockouts, you can run these on gels, you can use antibodies then to look for bands that change, and then cut out bands of interest and look for methylation sites and map those that change using mass spectrometry techniques. Equally, if you're trying to find methylated proteins, you can do immunoprecipitation, where you have antibodies that you use against lysates from yeast, followed by mass spectrometry. Or you can even take situations where you've got a proteome array, and we'll speak a little bit about this, where you've got around about 4,300 proteins on a slide, which you then probe with antibodies to find the proteins that are actually carrying methylation, subsequently which you can then do a validation of those sites using mass spectrometry techniques. Okay, so a simple kind of proof of concept. In this case, we have knocked out one of the lysine methyltransferases inside the yeast cell. And what we see is that leads to a loss of methylation of the band here. This is a Western blot that we are probing with an anti-methylation antibody. We can see that in the wild type version, we have methylation on this peptide at the last position here. Whereas in the knockout, where we've lost the band with our antibody, we now have an unmethylated peptide that's present. And we see a loss of 14 mass units associated with a loss of methylation on that peptide in the knockout. So from this type of experiment, we do a knockout of a gene, we look for a change or a loss in the signal that we detect with the antibody, and we then use mass spectrometry to find the site and the loss of methylation site associated with that. And so from that type of approach, we were able to do a whole lot of screening of methyltransferases inside the yeast cell. Here's just part of an experiment where we generated 32 knockouts of methyltransferases inside yeast. In this case, we've probed with a anti-dimethylation lysine antibody. And what you'll see is, of course, up here, we see the loss of methylation in this particular knockout of this gene inside the yeast cell at the time. And from that, we were able to find and show that there's this peptide here, which is dimethylated at this lysine residue. And ultimately, we're then able to confirm that this particular methyltransferase methylates the elongation factor 2 in yeast by doing a gain of methylation assay by expressing the protein recombinantly, the methyltransferase, expressing and purifying the elongation factor 2 and doing the gain of methylation assay. And so we named this enzyme elongation factor methyltransferase 2. To cut a long story short, here's another experiment which is similar, where we did more screening of methyltransferases, looked again and found, once again, loss of methylation on certain proteins. In this case, described the discovery of another enzyme, a new methyltransferase inside the yeast cell, elongation factor methyltransferase 3. These are actually very highly conserved, and just after we published this particular finding, soon after, the human ortholog for this was described by the Clark lab at UCLA in the United States. So these enzymes are very highly conserved in eukaryotes. And here's the discovery of yet another of these methyltransferases. This one is interesting. It's called elongation factor methyltransferase 7, and it methylates the N-terminus of the elongation factor inside the eukaryotic cell. And this is a completely new class of methyltransferase, once again, highly conserved. And in this case, Paul Falner's lab in Norway then discovered and described the human ortholog a few years after we described the yeast version of this particular protein. So very highly conserved. Now, so we've been able to do then some discovery of methyltransferases, and we've been able to find what we believe are the only substrates of those enzymes inside the eukaryotic cell. And for lysine methylation, it does appear that the lysine methyltransferases do only act on and do only methylate a single substrate protein. Okay, so they're very, very specific, those enzymes. They have very, very specific functions. But when you then go and look at arginine methylation, the situation, as we talked about a few minutes ago, is actually very different. In that with arginine methylation, there's a very small number of enzymes, but they can methylate a large number of substrate proteins. Okay, so completely different in terms of how the cell uses arginine methylation to how it uses lysine methylation. So given that we knew that there was likely to be a large number of substrate proteins for the major arginine methyltransferase inside the yeast cell, which is called HMT1, we decided to set up an experiment where we generated and used what's called a proteome array. Now we're very fortunate to be able to team up with Michael Snyder at Stanford. And what he'd already done for a series of different experiments was he had expressed and purified 4,228 proteins and then spotted them onto these array that you see here. These are actually just on a glass microscope slide. That's how these things work. And um, and what you'll see here is that after we've actually got the protein spotted onto these slides, we can actually then go in and probe with an anti-methylation antibody. And what they'll do is it will highlight proteins that are methylated with arginines. And so you might be able to see that wherever we've got two white dots, so we've got two white dots here, two white dots here, 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 and here, that's where we found arginine methylation to be present. So this is a very powerful approach because we've treated all of these proteins here, the 4,228 proteins, with the methyltransferase HMT1. That enzyme has methylated those which are at substrate. We've been able to detect methylation using the antibody, which are lighting up here, as shown as white. And so if you look at this, you can then kind of do a series of analyses of intra-array reproducibility or inter-array reproducibility. And from that, you can describe and find a series of candidates. Indeed, we predicted 43 potential substrate proteins of the arginine methyltransferase HMT1 from this experiment. But then you need to do the very hard work of validating that. And we validated in two different ways, whereby we did the same experiment, but then treated the actual proteome arrays with tritiated methyl groups. And here we can see a very strong signal in our proteome arrays coming up for protein NPL3. This has a lot of arginine methylation sites. So this is the treated version versus the control. And then there's other ones that had a weaker signal. So protein VAS1, we thought we had a signal there, but we couldn't, as compared to control, but we couldn't validate this with tandem mass spectrometry techniques after an independent treatment of the VAS1 protein with the enzyme HMT1. So this is a difficult experiment. We did find a series of candidates. We did confirm some of them, but many we could not confirm, leading to this kind of issue about have we found the right substrates for these proteins, or if we found some things which are false positives. So it brings us to this question about whether instead of using the proteome array as the means to try and find the substrate proteins for HMT1, whether we can instead use tandem mass spectrometry as the main method to find methylated proteins inside the eukaryotic cell. And so in this study, we described a large series of experiments where we did around about 368 tandem mass spectrometry runs on different preparations of the yeast proteome. We got really good data for around about 3,500 proteins. And from this, we did manual validation of around about 59 methylation sites, 35 lysine sites, 24 arginine sites on just 13 proteins. Okay, so smallish number of sites on a small number of proteins. And so this was quite good. We're quite happy with this, and we're very confident of the quality of the methylation sites we're able to describe here. However, what this study did reveal, and this has been a really challenging area of methylation biology and methylation proteomics, is that except if you're incredibly careful, what you can face is very high false discovery rates. And that if you're not careful, you can have false discovery rates that exceed 80% when you're doing mass spectrometry analysis of the proteome to try and find methylation sites. And there's a number of reasons for this. In part, it's because there are a series of artifactual alterations that can happen when they're exposed to methanol, to ethanol in the presence of acid. In part, it's an issue about there being small numbers of substitutions of amino acids that happen during protein translation, which we believe are contributing to the apparent appearance of methylation sites or mass differences associated with that. But you see that this is something which yeah, certainly got a lot of commentary from John Cottrell, one of the lead researchers in matrix science, the generator, the mascot search engine, and he wrote a really nice series of commentaries on this challenge of finding post-translational modifications on peptides, and in particular finding methylation. And he completely agreed with our kind of findings that you need to be incredibly careful to make sure that you get your methylation discovery right, so that you don't face this issue of false discovery rate. The thing that we described and suggested as really the most important thing to do for protein methylation discovery is to use what's called heavy methyl silac. And in that particular approach, what you do is you actually use a heavy methyl donor, so that when you actually put the methyl site onto your protein of interest, and this is inside the cell, you have a heavy methyl group which has now been added. And you can, of course, then do the comparison between a light and a heavy methyl group and its use and addition. And in a very similar way to how you measure and quantitate levels of proteins using just SILAC for doing comparisons of two particular samples to measure the quantities of proteins using tandem mass spectrometry. Now, what we did in association with the use of methyl silac is we developed new software and approaches to look for these mass differences between the heavy and the light versions of the heavy and light methyl sites. And then we also looked for the correlation of evolution profiles as these particular silac pairs come off the chromatography column during tandem mass spectrometry. And what we're able to do from that is to reduce our false discovery rate to about 0.53% in terms of the analyses. And then in subsequent studies, what we're able to do, and this is something that we just published relatively recently this year, was to describe now 82 methylation sites on 15 proteins, 113 methylation states. This study increased the number of sites of arginine methylation by 50%. And I think in this particular paper, what we're very pleased with now is that this paper, we think we've got an almost complete view on arginine methylation inside the eukaryotic cell. Previous studies in the knockouts that we did, we think we've got almost a complete view on all the arginine and lysine methyltransferases and their substrate proteins inside the yeast cell. And so what's important about this is that we now have this network that we've been able to build from literature data and from experiments that we've done and I've just presented to you. Now, we believe this is now a complete methylproteome network inside the yeast cell. We know all the methylation sites, we know all of the methyltransferases, we know the exact site where each of those methyltransferases actually acts. And not only is this a complete network for methylation inside the yeast cell and inside this eukaryote, we also think it's one of the few situations whereby we know all of the post-translational modifications on a group of proteins and all the enzymes responsible for that and the interconnections between them. So it now forms this really interesting kind of model system, if you wish to call it that, for doing systems biology research. Okay, it's got just enough parts to be interesting. There's 22 enzymes, there's 32 substrate proteins. And what I'm showing you here as well, these are the enzymes, these are the substrate proteins. I'm also showing you here the protein interaction partners of the proteins which are actually methylated. So we can now look at not just the enzymes and their substrate proteins, but also their partners, and try and understand the role of protein methylation in this protein interaction network. So you can see we're moving and have built this system, this small, compact, but really studyable and to a large degree tangible system to work with. For whereafter we can start to ask questions about this network, how it's actually put together and how it is regulated. And so for the rest of my talk, I'll really start to look more at this network and kind of investigate it in three different ways. I'll ask some questions about how we've learnt about the function of protein methylation from being able to look at this type of network. We're then going to ask some questions about how methylation is regulated, and then finally just explore some questions about how complex phenotypes of methyl mutants can actually be explained. And all of this will be done in the context of this network that I've just shown you and that we've been able to build from our experiments and, of course, from literature data, particularly from Steve Clark's lab and Pal Farner's lab. They're the two other major players in this space. Now, if you look at the ribosome, which is one of the proteins inside the cell, or a machine inside the cell that has a lot of methylation sites, you'll see that methylation is largely found at protein surfaces and also in some cases at protein interfaces. And this is a similar type of analysis that we did in 2018 when we were specifically looking at elongation factor, which is, of course, something that interacts with the ribosome. And once again, you see the same thing. So look how the methylation is at interfaces between elongation factor and parts of the ribosome. And here you see it again at the same site in another orientation. So this raises this really interesting question about, well, is it the case that protein methylation, its function is actually to regulate protein-protein interactions, or even is its function to regulate the specificity of interactions that can occur for proteins? And these are the types of things that we've spent quite some time investigating with the knowledge that we have proteins that are methylated, and we know, of course, the methyltransferases that are methylating them, and also know the proteins that are interacting with the methylated species in the first instance. So we wanted to explore this question: does methylation regulate interactions or regulate interaction specificity? So to do this, we built a new two-hybrid system. So you're probably familiar with the two-hybrid system. It's a way by which you can express two proteins of interest inside a cell. And if those two proteins do interact with each other, then it turns on a reporter gene. And so usually what happens is you do that, you express your two proteins of interest. Here we've got a gold protein, a green protein, and they're not interacting in this situation. So the reporter gene, in this case adenylate cyclase, is not turned on. But what we did is we decided to then co-express a methylation enzyme, so that we've now got a methylation enzyme inside, in this case, the E. coli cell. It's methylating a protein. It happens to be a yeast protein we're expressing here. And this methylation, if it does bring about the interaction with another protein, we then see this reporter gene being turned on. So we're basically asking a question: does the presence of methylation on one protein, in this case here, modulate or bring about an interaction between it and its partners and turn on a reporter gene? So we're looking for modification-dependent interactions using this two-hybrid system. And so this is one of the first questions that we asked: does lysine methylation by the enzyme CTM1 affect the interactions of cytochrome C1? And so here's a different view on the network that we were looking at the time. And we said, well, here's the enzyme, cytochrome methyltransferase 1. Does the methylation of cytochrome C1 affect the way in which it interacts with these two proteins that we see that are the interaction partners it has in this network? Now, we were able to show that the methylation inside this two-hybrid system we developed was successful. Here we see the trimethylation event being recapitulated inside E. coli as part of this whole assay. And then we were able to do the measurements of interactions using this conditional two-hybrid system. And I guess the main point that I want to show here is that when you do add methylation onto the Psych1 protein, you get an increase in the amount of interaction between Psych1 and either the ERV1 protein or the Psych3 protein. We see an increase in the amount of interaction that occurs when it's methylated as compared to when it's unmethylated, either with the dead enzyme control or just the no enzyme control, the no methyltransferase control. What we did see, however, which is interesting, is that when we methylated inside this assay protein CCP1, which is another interaction partner of Psych1, it did not increase the interaction that it had with Psych1. So what we're starting to see here is that the methylation was increasing some interactions, but having no effect on others. Okay, so it's hinting that there's actually interaction specificity, which is being regulated by methylation inside the eukaryotic cell. Now, we wish to explore that in further detail. And here, of course, we're going back to our major arginine methyltransferase. It's HMT1. Inside the human cell, it's called PRMT1. It has very much the same type of function. And we wanted to explore the role of the HMT1 methyltransferase in a network and see here we are looking at one of the substrate proteins of HMT1 called NPL3 and asking the question, does the methylation of NPL3 affect its interaction with these 5 different partners inside the network? So of course we did all the molecular biology, set up our 2 hybrid system, and then did the assay. And we're really asking, well, does the methylation of NPL3 affect the specificity of its interaction? And here we see a series of protein pairs which we are monitoring. And this is without methylation being present inside the assay. When we add the methyltransferase, in a number of cases, we saw a significant increase in the amount of interaction that occurs. And then if we did the same assay with a dead enzyme control, we see how in many cases it just kind of comes back down again. So we did see a significant increase in interactions in the presence of methylation in some cases. Other cases we saw no or little increase at all. So once again, suggesting that methylation is important for some interactions of NPL3 and indeed a requirement or certainly something that assists greatly, but in other cases that's not required at all. But NPL3 as a protein has more than 20 protein-protein interactions. Does it have an interaction code? And this is what we'll just spend a few minutes more talking about. Now, protein interaction codes, we were inspired to write this because of the existence of something called the histone code, where histones have a large number of different modifications, including methylation, but also acetylation, phosphorylation, that changes the type of interactions that those proteins actually have. So in this paper, we presented it's really a generalized hypothesis about protein interaction codes, whereby post-translational modifications affect the specificity of protein-protein interactions inside the eukaryotic cell. And there's a number of ways by which this can work, and I'm not going to go into this in detail. In some cases, the modifications can affect the interactions that a protein may have, the stable interactions. In other cases, post-translational modifications may affect the interactions with other modifying enzymes. Okay, and we'll talk a little bit about that in just a minute, because this is the basis of what's called post-translational modification crosstalk. Now, to kind of explore this kind of question about protein interaction codes and explore it for the arginine methylation, what we noticed is that there's these SRGG motifs which carry methylation. So here's methylation occurring in this SRGG motif. Here's the spectra for this. And there's actually only 3 proteins inside the yeast cell that have the SRGG motif in tandem repeats. Okay, so there's 3 of these. Now they're functionally related. They're in the nucleus. They're actually all functionally associated with the processing and transport of messenger RNAs. Now, what we also noticed and indeed showed is that when you have any of these RGGs, that the serine that's here can also be phosphorylated, and we showed it's phosphorylated by the kinase Sky1. Here we're just showing those phosphorylation sites, and here's the spectra associated with that. So we wanted to ask the question, do the modifications that we see in this SRGG motif, do they show post-translational modification crosstalk? So is it the case that if you put methylation onto the protein, does it then block the addition of phosphorylation? Or if you put phosphorylation onto the protein, does it block the addition of methylation? You see this interplay that exists between these modifications. This is a very fine version of control of proteins inside the eukaryotic cell. And so just to kind of jump over, I guess, quite a lot of work which involved use of peptides, modified peptides, and treatment with recombinant enzymes. There's two things which we were able to show. One is that when you have phosphorylation on a serine in the SRGG motif, this blocks the capacity of the HMT1 to add methylation to the arginine. So phosphorylation blocks methylation. That was a very clear rule that exists for this type of crosstalk. But methylation does not block phosphorylation. If we have methylation here in the arginine, the SCY1 kinase can very happily add phosphorylation onto this particular motif. So it's not a bidirectional crosstalk, it's a unidirectional crosstalk that exists here in this really kind of interesting regulatory system. So we're saying that, well, the phosphorylation blocks methylation, but the methylation does not block phosphorylation. But then the question that we had was does this actually regulate or affect protein-protein interactions that NOP1, which is the protein subject to this phosphorylation methylation, actually occurs? So is there this specificity of interaction that occurs as you phosphorylate or methylate the proteins in terms of how NOP1 can interact with its other partners? So how can we investigate this? So once again, we did some more of these types of conditional to hybrid assays. And what we were able to show is that the methylation of SRGG increases interactions of the NOP1 protein with three other interaction partners. So this just shows how you get an increase in the methylation. This is the increased level of methylation that we see, of interaction that we see when the protein is methylated as compared to unmethylated. We see that again and again. But phosphorylation within this assay did not increase the interaction of this protein with any of its partners. So there's something going on here. And the question was, of course, why does phosphorylation not change any of the interactions, whereas methylation does change the interactions? And so this is the model that we came up with, in that you've got the protein NOP1, and what it has to actually do is it has to get inside the nucleus and then become part of the nucleolus. And what we believe is happening is that the protein NOP1 is actually initially phosphorylated by SCY1 in the cytoplasm. This allows it to interact with a CAP121 transporter to actually bring it inside the actual nucleus itself, whereby GCN5, a phosphatase, and we've shown this inside this paper, removes phosphorylation. And then once the protein is dephosphorylated, it can become methylated, and the methylation then stabilizes the interactions of its partners to form this functional complex, which ultimately is involved in the processing of ribosomal RNA. So the reason why it doesn't get methylated out here is that if it did get methylated out here, it would interact with partners inside the cytoplasm and make it impossible for it to get transported into the nucleus. So we think the phosphorylation and methylation is a means of regulating the import of the NOP1 protein into the nucleus, and a means to make sure it does not complex with its partners in the cytoplasm, but only does that actually once it reaches the nucleus. And just to show that this actually is something that happens, here's some microscopy that's done with this experiment. And we show that when we disrupt the methylation of the NOP1 protein and disrupt the methylation of that SRGG motif, we disrupt the formation of the nucleolus. That's why we're seeing this speckled nucleolus here in green as compared to the wild-type nucleolus, which is what we see over here. So the loss of arginine methylation in the SRG motif disrupts the formation of the nucleus, of the nucleolus. Okay, so just in the last few minutes I've got, I'm going to talk a little bit about, I guess, two final things. One is just the question about how is methylation regulated, and the other is how we can explore complex phenotypes of methyl mutants. So methylation is, of course, something which is relatively widespread. It's on a lot of histone proteins. And one of the things that's interesting and we've been exploring for some time now is when you look at the histone proteins inside the yeast cell, there's 8 enzymes that are associated with their methylation and their demethylation. And so we thought this would be a really nice, once again, small but tractable network to work with to try and understand how methylation in a small system is itself actually being regulated. So here I'm showing you the 4 methyltransferases that can act on the histones. These are the 4 demethylases inside the yeast cell that can remove methylation from the histones. And so we're really quite interested to use this as a means to study, as I mentioned, the regulation of methylation inside this small system. And in part, one of the reasons why we think it's such a unique thing to study is that histone methylation exists in really kind of intricate patterns. And here I'm just showing you, here's the methyl mark. And here I'm showing you the patterns that exist of those methyl marks along different genes. And you can see there's really intricate and remarkable control about how much methylation, where you've got a lot or not so much, occurs actually along the body of genes. So this has to be regulated somehow, and it has to be involve the methyltransferases and demethylases. So here we just ask the question, well, are the methylation enzymes phosphorylated? Is it phosphorylation which is regulating them in their activity? And I suppose to just give you an overview of what we've done here and what we found is that we completely characterized these 8 methyltransferases and demethylases for phosphorylation. And from this, we've described about 75 phosphorylation sites on the methyltransferases and the demethylases. And we see certain patterns in that some demethylases, for example, have a lot of methyltransferases, whereas other demethylases do not. We see that most of the enzymes are quite heavily methylated, whereas, as I mentioned over here, the demethylation enzymes, some are heavily methylated and others are just not. So the phosphorylation is being used differently per enzyme in this system of 8 enzymes and the 4 histone proteins. And the interesting thing is that when you look at some of these methyltransferases that act on the histones, the set1 and the set2, we see some of the phosphorylation sites are actually conserved in terms of the amino acids. And we actually see some of the phosphorylation sites themselves have been shown to be conserved all the way from yeast through to man. So we're seeing regulatory processes inside the yeast cell that are likely to be used inside the human cell for the same purpose, which is the regulation of modification of histones. Now, the phosphorylation of methyltransferase, it's enriched in disordered regions. You can see this very clearly. These are disordered regions. These carry phosphorylation sites. And in particular for the methyltransferases, we kind of see that. But we don't see that for the demethylases. Okay, so the phosphorylation sites in the demethylases are in different parts of the protein, whereas in the methyltransferases, look how the phosphorylation is always found in these disordered regions of the protein. So we reasoned that, well, when you find phosphorylation on these disordered regions, they must be functionally important. And so we did some mutagenesis analysis here to try and figure out whether the modification of this enzyme then changed the way in which it is methylating. Okay, so does phosphorylation of a methyltransferase affect the way in which it methylates, in this case, a histone protein? And so we changed these amino acids here. There's the start of the set2 protein. And when we changed it to alanine, and so reduced the negative charge at the start of a set2, we all of a sudden saw a dramatic loss in the amount of methylation present. So we saw much more of the unmethylated, this is all unmethylated here, rather than methylated versions of the histone proteins. So phosphorylation of the methyltransferases is affecting the way in which they are then methylating the histones inside the eukaryotic cell. And we now have a regulatory network that we've started to build, where we have the histones, we have the methyltransferases and demethylases, and we're now starting to map kinases onto the methyltransferases and demethylases to build a regulatory network of histone methylation. This is work in progress because the business of kinase discovery up here is very, very difficult. So these types of links that I'm showing you here are actually predicted at the moment, apart from a few. Whereas in this part of the network, all of these relationships between the enzymes that methylate and demethylate the histones are known and are very well proven. So our goal over the next 5 to 6 years is to build this level of the network, so we understand how the histone methylation sites are being regulated as part of that complex pattern of epigenetics inside the eukaryotic cell. Now, just in the last couple of minutes of my presentation, we'll just explore a little bit about this question about complex phenotypes of methylation mutants. And our complex phenotypes are things which are found inside the eukaryotic cell. You can knock out or have a change in one amino acid of one protein, and you can see many unrelated phenotypes in some cases. This thing is called pleiotropy or pleiotropy. And it's one of the things that you see in human disease, and you can have unrelated phenotypes which come from a single mutation. And part of the question has always been, well, how do complex phenotypes arise? And in this case, for our methyl mutants, can this be explored? And I suppose our case study here is we're looking at there's what's thought to be just a single histone, histidine methylation event inside the yeast cell at ribosome protein RPL3. It occurs just in this part of the ribosome in here. There's different phenotypes. There's elongation defects in the ribosome. There's perturbation of ribosomal subunit levels. And what we also observed, and this is work done that's unpublished by our lab at the moment, is we did a whole series of screens for sensitizers. These are standard phenotypic screens for yeast, and found that when you knock out this single histidine methylation site in a ribosomal protein, we all of a sudden have sensitivity to hydrogen peroxide, to amphotericin B, cold sensitive versus what you see at room temperature. So complex phenotypes are arising not only in protein translation, but also a number of other things that you see inside the cell. So if you do standard proteomics, then you can use that and you can find proteins that are changing. And this is just an example of this. We see that there's proteins that are downregulated on knockout. There's one that's upregulated on knockout of the HPM1 enzyme. So that loss of a single histidine methylation site can have quite an interesting range of effects. And some of these are interrelated. We see that the loss of the trehalose pathway or downregulation of the trehalose pathway is almost certainly associated with the cold sensitivity that we see in the knockout. But this didn't explain all of the phenotypes that we were seeing on knockout of HPM1. And so the question that we had is, can we use other types of proteomic techniques or technology to try and understand these complex phenotypes that we are seeing on knockout of this methyltransferase, but not being able to explain with expression profiling, which is what we've done in this proteomics experiment just here. And so for this, we've used cross-linking mass spectrometry. And for those of you who may not be so familiar with this, what you do is you start off with a lysate of proteins from a cell. In this case, we're going to be working with whole yeast cell lysate. And we've done protein cross-linking, and cross-linking can both generate cross-links inside proteins, but also between proteins, which is what we see here. You can digest those proteins into peptides, and what you'll have is a mixture of uncross-linked peptides, which is what we see here, and also cross-linked peptides, which is what I'm showing you here. Now, of course, in some cases, those cross-linked peptides, like this one here, actually represents a protein-protein interaction. And this is what we're most interested to be able to see. Can we detect and quantitate multiple protein-protein interactions inside the yeast cell? And can we compare the interactions between our wild type and our knockout, and then find things that may explain the phenotypes that we've been observing? So technically what you do, there's an enrichment step. We've used strong cation exchange. And there's then 3 rounds of mass spectrometry we typically use to measure the mass of the cross-linked peptide, to then break the peptide into 2 pieces. And then we do independent fragmentation of each peptide, and then use that data to identify the peptides that have been cross-linked. Okay, so this is just a bit of a view on our results so far. So from this, you can detect a lot of cross-links. Okay, so there's 8,400 cross-links being detected here. This maps down to around about 1,267 cross-links, and of these, we actually have around 261 inter-protein cross-links that we generate, and these are allowing us to measure 146 protein-protein interactions at the same time in one experiment. So it's quite a nice kind of thing to be able to do with this type of technology. And you can generate a network of protein-protein interactions from your cross-linking data, which is what I'm showing you here. But the more important thing for us was, can we actually detect changes in protein-protein interactions? And with this, we coupled the cross-linking mass spectrometry with the SILAC-based approaches, did our data analysis, and from this, we're able to find a reasonable number of crosslinks that we could quantify in both replicates. And from that, we're able to describe a number of situations whereby we're seeing a change in the amount of crosslink occurring between wild type and the knockout. So this is the knockout, this is the wild type. But this is actually happening where there is no change in the amount of protein present. Okay, so here we've measured the amount of protein. The ratio of protein between wild type and knockout remains 1:1, whereas what we're seeing is a change in the amount of protein-protein interaction between the two proteins that we're seeing here as part of the TIM complex for the import of proteins into the mitochondrion. So we're seeing these types of situations, and we believe that some of the changes we see in protein-protein interactions that are occurring here are probably associated with the sensitivity to hydrogen peroxide that we're seeing here, because this is, of course, associated with and known to be caused by changes in the in mitochondria and the respiratory chains that exist there. This is just a larger level view on this in terms of, here are some of the changes that we see in protein-protein interactions that are shown in color here. Anything in color is a significant change in interaction. But these are happening in situations where the amount of protein, okay, because this is protein, this is the interaction. You see most of the proteins here are not changing, but the level of interactions between them is changing inside this system. Okay, so just to conclude, so I've taken you on a bit of a journey of protein methylation. Now, it's a modification which is increasingly well described and well understood, and particularly so in yeast, where we believe we've now constructed, with data from the Clark lab, the Founders lab, our lab, a complete network of protein methyltransferases, their substrate proteins, and in many cases, we know what the interaction partners of those are. Now, the function of most sites are not understood, but we know that methylation can modulate protein-protein interaction, so that's very important. In terms of how methylation is regulated, we've really explored two things, and I've given you two examples. One is how methylation can exist in crosstalk with protein phosphorylation, that competition that you can have on the same protein. We've also shown that methyltransferases can be regulated by phosphorylation with that histone example. So now we've got these really fascinating questions. Can we connect the histone system, which is what we've built, to the cell signaling system? Because there's a gap at the moment that exists between how the epigenetic system works, how it's methylated, and then the signaling system that actually exists inside the cells. And that's one of the major goals that my laboratory has over the next 5 or 6 years. And finally, I've just shown you some of our work with cross-linking mass spectrometry as a way to monitor many interactions in vivo. It's an in vivo cross-linking that we do in a single experiment. And we believe this may provide a major new means to explain complex phenotypes. So this is just some of the fantastic team, not all of the team that have contributed to the work that I've shown you here today. We're very fortunate to have a fantastic group of people to work with. And on the next slide, here is the more formal acknowledgements. People that have made substantial contributions include Josh Hamey, Jean Hart-Smith, Ignatius Pang, Melissa Ursay. Ryan and Tara have also made very significant contributions to the work that I've shown you here today. I'd like to acknowledge the mass spectrometry facility at our university, Snyder Lab, Mike Snyder in particular at Stanford, James Bruce and his lab at University of Washington, and the funding bodies that have made this work possible. And once again, I thank you for your attention, and I'd be really, really happy to take any questions if they may arise. Thank you.
Thank you. And thank you, Professor Mark, for the very insightful and sharing with us about the maturation and from the very beginning of introduction to and bring us into the detail of how maturation in the regulation and crosstalk even with the phosphorylation, as well as showing a few examples to see how cell actually regulating in this case using a methylation between protein, and bring us also to the network at for the group of the protein may be crossly regulating by the maturation, for example. So bring us into a idea that those protein may be having a similar function and kind of regulation in the cell that nicely presented. So thank you very much again. I learned a lot from your lecture here, Mark. So we are open for the floor to ask questions, and we currently having a couple of questions in the chat box. And all the participants, you are welcome to type your question in the chat box. And so I will start with Q&A with a question from Dr. Go, also a research fellow in INBIOSIS and secretary for Nation Proteomics Society. His question is how the candidates were selected for knockout screen. In the knockout screen, how the candidates were selected.
Yes, no, thank you so much for the question. So, what we know inside the yeast cell and the eukaryotic cell is there's three major domains that you'll always find inside methyltransferases. The most important of them is the one which actually binds the methyl donor, which is ADOMET. So what you can do is you can search for proteins that have the ADOMET binding domain. That's a well-conserved domain. It's in all methyltransferases. And that will give you a list of methyltransferases in total. But then as you mentioned and you asked, the question is, well, how do you know which ones are protein methyltransferases or DNA methyltransferases, RNA methyltransferases, things that act on other small molecules? And part of the challenge is that you don't actually know. So what that did mean is that we had to do a large number of screens of the methyltransferases just by knocking out. I think we ended up looking at about 50 or so methyltransferases, either building the knockout ourselves or getting them from the knockout collection in yeast. When we did our CRISPR-Cas9 knockouts for the human cell, they were much more targeted in that we'd already found a methyltransferase and confirmed it inside the yeast cell, and then we were looking at the ortholog for the mammalian knockouts. So unfortunately, there's hard work to do with the screens at the beginning, but once you've found them in yeast, because yeast genetics is it's actually very simple and relatively straightforward. Once you've found the enzyme in yeast, then of course you can look in a much more targeted way inside the mammalian cell, and that's been quite successful.
I see. Thank you, Mark, for sharing. We have another question from Leong. So does the methylation of lysine and arginine reduce the efficiency of tryptic cleavage? If it does, which of these, example, mono, di, and trimethylation affect the most? So would the methylation actually influencing the cleavages, I think, during
Yes.
perhaps?
Yeah, yes, no, of course it does. You're absolutely right. And it's one of the things that sometimes people use to stabilize proteins is to chemically methylate them. So it does affect the cleavage, and trimethylation affects cleavage most strongly. It almost blocks tryptic cleavage. So it is one of the other signatures you can look for when you're doing protein methylation studies, is if you've got a peptide that's quite long and has a methylated lysine in the middle, then that gives you some confidence that it's a true methylation site, because trypsin doesn't like cutting at lysines which are methylated. With methyl arginine, trypsin is not quite so fussy. It doesn't seem to mind as much whether you've got methylation, but certainly when you have methylation on lysine residues, trypsin doesn't cut quite so well. But it's useful in some respects, but it does make things complicated if you're trying to do quantitative analysis, because you can have a peptide and you can have it in an unmethylated form, a monomethylated form, a dimethylated form, a trimethylated form, and you can all of a sudden now have to try and quantitate 4 peptides to try and get a measure about how much peptide is present. So it does make things complicated, but yeah, certainly it does change the way that trypsin works.
So that making it complicated in terms of like when doing the mass spec. So more mass spec have to be done, or how to overcome actually the.
Yeah. Now look, it's a good question. One of the main complications is if we're trying to compare the amount of methylated peptide to the amount of an unmethylated peptide, because if you have a methylated peptide, it may have a missed cleavage because the lysine is got the methyl group. So can be quite a long peptide because it has missed cleavage, whereas the unmethylated one will be smaller because trypsin is cut at the site which you've got methylation. So it just makes it complicated in terms of how you do a comparison and really compare the amount of methylated peptide, which can be very long, with unmethylated, which can be quite small because it's that small part of the peptide where trypsin is actually cut where there's no methylation.
Right. So followed by that you as well the question, given that the same site on the same substrate can be methylated by multiple methyltransferase, would that affect the results of KO screen, which was based on the knockout of a single methyltransferase gene at one time?
Yeah, thanks for that really excellent question. So I guess there's 2 answers to this question. So the first answer is an answer I can give you for the yeast cell. So what is really good inside the yeast cell is that do we have an exception here or not? I'm trying to think. I think I can say that in the yeast cell, where we have the methyltransferases that act on a single substrate site. In yeast, there is only one methyltransferase that can act on any one site inside the yeast cell. So I think we've now shown that you can only have one enzyme acting on one site. Whereas when you start to then look at the human cell, things become much different, and in particular for histone methylation. Because for histone methylation, as I showed at the start of the presentation, there's more than 50 different methyltransferases that act on a very small number of methyl sites. So the knockout analyses that you can do in yeast, look, it's just fantastic because you've got a single enzyme acting on a single site in a single protein for all of the lysine methylation sites. The only exception there is that there's actually a couple of situations where there's some paralogs in the ribosome, very recent paralogs that have arisen. So you've got one methyltransferase that acts on 2 paralogs of the same protein inside the ribosome. But certainly, I mean, it does get very complicated inside the human cell, and so the knockout screens are much more difficult. But we believe inside the yeast cell, it's gorgeous because we have this one-to-one mapping for all the lysine methyltransferases in the site, and then for the actual arginine methylation, there's one major arginine methyltransferase inside the yeast cell that acts on many proteins, and then there's 2 or 3 others, but once again act on single sites. So yeast is fantastic as a model system for this type of thing, but it does get much more complicated inside the human cell, where you do see that redundancy, where you can have multiple methyltransferases acting on the same side.
So followed by this, Mark. So when you are sharing about a single enzyme and single substrate protein site, and followed by with a question from that you here.
Mm.
So it is not surprised that we would have multiple enzyme to one substrate or kind of a reserve system, or because in terms of a security or the economically, you would want to have one enzyme maybe at two different job, right? And also to be safe that not only one particular enzyme that would um, do all the job. If you have a problem, then you may have someone to replace and do and carry the job to let the cell to live. So would that taking in consideration in during the like analyzing the results, as just now like your question is would the other than material transfer would likely exist, right, to rather than a single enzyme, a single protein. No, I just found it fascinating because it means that if it's a single enzyme, single substrate, it would be very important for that particular system that to having a dedicated a protein that modify it.
Yes, yes, yes. No, these are excellent points, and certainly what we see with lysine methylation, and if we talk, predominantly about yeast at the moment, because it's a simple system, but what we see in yeast is almost universally conserved. So when we look at all the lysine methylation inside yeast, um, you'll see almost all of the methylation is actually on ribosomal proteins, or is on ribosome-associated proteins. And what we do see is that if you knock these things out, functionally, there's actually relatively weak phenotype in most cases.
Mhm,
Um, but what we do suspect, and I think maybe the most important case in point is actually there's a protein called elongation factor 1 alpha. It's
mhm,
a really important protein. It takes charged tRNAs and brings the amino acids into the ribosome.
mhm,
And this protein it actually has 5 different methylation sites. It has
mhm,
5 different methyltransferases that act on it. So just leaving apart the histones for one moment, this is the most carefully methylated protein inside the eukaryotic cell. But when you think about it, it's actually one of the most single important proteins in the eukaryotic cell. If you can't bring amino acids into the ribosome,
mhm.
then you can't grow.
Yeah.
At the same time, it's the type of thing whereby if you make a very small change to the protein in its structure and you marginally increase its efficiency, you'll increase the rate at which the cell can grow.
Mhm,
Because it's making protein. It's actually the most important step in terms of bringing amino acids into to the ribosome. So what we believe is going on there is that these lysine methylation events, the cell uses extremely carefully to fine tune, in particular aspects of protein production in translation. And you can
right.
see how once, of course, they give you a tiny evolutionary advantage, that you're more efficient or more accurate in making protein, very
Mhm,
quickly that will become fixed into a population and become part of the genetics of future species. So we think that there are the reason why these specific methyltransferases exist, at least in the protein translation mechanism and machinery, is because it does confer evolutionary advantage. And when you look at the methyltransferases and their lineage, one thing that's fascinating that we see in elongation factor is there are these 5 different methyltransferases, and they each actually have independent evolutionary lineage.
mhm.
So it means it would have evolved over a long period of time, but you can see why they're there because they just change the efficiency in a small but favorable way of protein translation. When you can make protein more quickly or more accurately inside a eukaryotic cell, of course that's going to make you much more competitive than an organism that cannot. So there's really kind of very fine changes that are
Thank you.
happening, but you can see why the cell has kind of invested in them and why they've become fixed inside the eukaryotic population.
Right. Thanks, Mark, for this
You're welcome.
explanation. So we have a question from Professor Naslan. Can methylation be used to change the physical locations of the proteins and their interactons within the cells? Is there any evidence of this, or can this be investigated?
Yeah, no, look, that's a fantastic question. Yeah, yes, there is evidence that methylation is important for the import of a number of proteins into the nucleus. Sorry, it's actually the export of a number of proteins from the nucleus. Um, so this is methylation of arginine, which is known to be responsible for this, and you see it in a protein called nucleolar protein 3. And nucleolar protein 3 is interesting in that it's a shuttling protein, so it has to go from the cytoplasm into the nucleus, and then it needs to take messenger RNA and export that from the nucleus. And what's known is that it needs to get phosphorylated in the cytoplasm to then go into to the nucleus, and then it needs to get methylated inside the nucleus, and then needs to grab messenger RNA, and its methylation is essential for it to be then exported. If you don't have methylation, then you don't get the export of that protein from the nucleus. So certainly for the shuttling of proteins and certainly between the cytoplasm and the nucleus, we think that there's a role, and we showed this with the other nucleolar protein, which is NOP1, nucleolar protein 1. And we also see it with protein NOP3. NOP1 is fibrillarin inside the mammalian cell.
I see. Thanks for Mark. From here, I also have further from here because you share with us that one of example that protein to be should be methylated in the nucleus. Otherwise, then it would maybe complete with the is other protein to
Yes,
find a
yes,
partner.
yeah.
So that brings us a question where are there a certain group of methyltransferase is active in cytoplasm and some are active in nucleuses? And with that, is that again a signal or modification is needed for those to be maybe active in the nucleus?
Yeah, look, fantastic question again. So certainly there is, and there are some of the methyltransferases that are only found in certain compartments. So we do know, of course, that the ribosomal methyltransferases are largely cytoplasmic in terms of their localization. We know that the major arginine methyltransferase, which is HMT1 or PRMT1 inside the human cell, is found inside the nucleus. There is, and yeah, I'm just trying to remember. So the mitochondrial methyltransferase, CTM1. Oh, gee, I've actually forgotten if that's inside the mitochondrion or whether it's inside the cytoplasm, that one. But of course, I mean, it's something that has a very specific role, which is important for
Mhm.
the modulation of the cytochrome C1 with its interaction with Cyc3 and ERV1. So there are certainly some that are found in certain parts of the cell. And their roles are also clear in terms of the proteins they interact with and the things that they then do. So we see that, and I think in yeast it's quite clear. Inside the human cell, it's much more complicated because you've got a very large number of methyltransferases,
Mhm.
and you're likely to have some that I think have more of this diverse cellular localization.
Thanks, Mark. So we have an interesting question here about, have you observed any methyltransferase to methylate both DNA and proteins? The question from that you.
Oh, gee, look, this is a fantastic question. So, and what do we have here? I mean, we certainly have a protein which is methylated, which can then itself methylate nucleic acids. But I don't think there has been a documentation of a protein that has a dual substrate. Um, so I don't think there are any methyltransferases that are known to methylate a protein and methylate a nucleic acid. But as I mentioned, there is it's actually protein fibrillary, which itself is a methyltransferase, but it's methylated by HMT1 or PRMT1 in these very unique and extended SRGG domains. So that they are
See.
very specific these methyltransferases, and certainly in most cases they have a much higher specificity, we believe, than the many kinases that do have multiple potential substrates and a lot of overlapping specificity.
Mm. So far we haven't found one then.
That's right. Yes.
Right. We have another question from Charles Alvin Saturalam Walavan. The question is, may I know if methylated protein can be used as a biomarker for certain type of disease?
Yeah, I think the answer is yes, in that there are certainly some aspects of histone biology and epigenetics, which are of very high interest in the field of cancer biology. And there are, of course, some drugs that have been developed against demethylases, in particular the demethylase LSD1, which is either in clinical trial or now on the market for cancer treatment. Equally, there's been quite a lot of work, particularly done by Tiziana Benaldi in Italy, to look at arginine methylation, the way that arginine methylation on proteins is associated with and changes as part of cancer. So yeah, my laboratory doesn't work on human disease, but I think there's some good examples in the literature and some quite extensive studies and really excellent reviews which are now written. Um, um, by a number of different groups about the association of arginine methylation, lysine methylation with disease, and of course the question being how you can have are there biomarkers? I think the answer is definitely yes.
Yes, I see. Let's bring to another question that linked to this from Hakim Ransak. Do you see possible therapies utilizing methylation or demethylation method
Yes.
for therapeutic method?
Yeah, I think I'd give a similar answer. We certainly see that, and one thing that we find is and think is really important with methylation and as much as anything else the methyltransferases is that we know that the methyltransferases are enormously specific, and so that means that they make really interesting drug targets because they act on in most cases these single targets for the lysine methylation. So it means that if you manage to design a drug against that methyltransferase, you'll have something of very high specificity. Okay, so that's a really kind of important point. The other thing which we think is very important is that the methylation is in many cases obviously being used to control protein-protein interactions. And so that gives you, a kind of a very nice, switch that you can potentially use when you have methylation there or not there. And so if you can control the methyltransferase, or in some cases the demethylase, then you can control that switch, and then you can start to control interactions of proteins that may be of high biological importance. So we think they're enormously kind of interesting enzymes, the methyltransferases, the demethylases. And there's been quite a lot of literature written about how important they can be or should be for those reasons that we've just been discussing.
Thanks, Mark, for that. I think we have like last two questions, Mark. So
Yes, I
there's a
think these are
question.
fantastic questions. Thank you so much, everyone, for sending them through.
So we have a question: how far methylations can influence the identification of proteins? A question from Losmawati.
Yes, I think that methylation usually doesn't really affect the identification of proteins. I guess there's some situations where it may affect your capacity to find parts of proteins. And what I'm thinking of here is that there's some parts of proteins that have a lot of repeats, some of these tandem repeats that I described of SIGG, SIGG,
Mhm, mhm.
SIGG, SIGG. And in those situations, when you have methylation at every arginine residue, there's a few problems there. I mean, in part, either trypsin can't cut because there's methylation everywhere.
Mhm.
Or in other situations, if you have less methylation, then trypsin cuts everywhere, and you've got these really small peptides that become
Mhm.
hard to identify. So what we have done is for some proteins with a lot of methylation, we can't use trypsin to characterize those regions, but we tend to use chymotrypsin
Mhm.
because those proteins tend to have uh, a lot of tyrosine in them, sometimes phenylalanine. So we tend to use 2 or 3 enzymes if we're trying to do detailed characterization. We use trypsin, chymotrypsin, sometimes lyc, sometimes asp-N, if we're working with some of these methylated proteins, just to make sure that we're not missing things.
Okay. So we have another question before the last question.
Yes, thank you.
That many cross-linking reagents that are widely used have active groups that react with lysines. Would that affect the types of cross-linkers that you choose for methylation-specific cross-linking experiments?
Yeah, no, look, that's an excellent question, and it's something that we've been working through and trying to understand ourselves. Because you're right. I mean, yes, if you have a methylation on a side chain, it reduces its capacity to cross-link. So whenever we've used these types of cross-linkers in methylation experiments, I think the only studies that we've published of that type have actually been where we've been looking at arginine methylation. So a study that we published, I think it was the start of last year, where we looked at the activity of the arginine methyltransferase on one particular protein, and we sought to capture a transient protein-protein interaction with protein cross-linking. So in that situation, it was a arginine methylation rather than lysine methylation. But there's really kind of nice sets of cross-linkers that are available where we're really interested in, and we've been using a cross-linker called DMTMM lately, and that's a zero-length cross-linker. It's also a very fast cross-linker. We like to do in vivo cross-linking because that way you can capture interactions as they're occurring inside the cell. And so we very much like DMTMM. We've been using, and we're very fortunate to have a collaboration with James Bruce to use his PAR cross-linker. That's lysine reactive, but it is one that we can get inside the cell, and of course DSSO does work in terms of getting inside the cell, but of course that's a lysine specific cross-linker. So absolutely, I think your point is an excellent one, that you need to think carefully about what you're studying and choose the right cross-linker before you do the experiment.
Okay. Thanks for that. And we come to the last questions from Hakim Razak. What future questions do you have you think would arise due to protein-protein interaction within other fields?
Oh, look, these are, yep.
Yeah, yes, please.
Yes, these are fantastic questions. The things which we've been thinking about and focusing on a lot are the ways in which we can build networks where we know all of the members inside those networks. And so the two that I've shown you today, one is of course the methylation network, and the other one, which has got a similar amount of detail, at least for methylation itself, is the histone network and the histone methyltransferases. So what we're very much doing is we're building these small networks. Then they're not tiny, but they're important. And our goal is to fully characterize the role of methylation in those networks, but then fully understand how those networks are regulated. And this is when, of course, we need to understand the methyltransferases, how they actually connect to the signaling system inside the cell. Because, of course, when you're looking at methylation, it's one level of regulation on protein-protein interactions, but the methyltransferases are always, I think we can say this, always phosphorylated. And so there's a link to all the signaling system inside the cell, which is ultimately feeding into these things and controlling how and who they regulate. So the protein-protein interactions that we look at and believe are important, there's actually different types. So we're very focused on, of course, the way that protein-protein interactions themselves are controlled by methylation on this protein, for argument's sake. But then, of course, the methyltransferase that puts the methylation onto that, we're very focused on how that methyltransferase is phosphorylated, and that, of course, involves an interaction with a kinase, which is upstream. So we look at interactions in different ways. Some of them are transient, some are associated with signaling, but we're very much focused on looking at important parts of the cell, such as the histone methylation process, and completely characterizing that in terms of, well, how does the phosphorylation affect the methylation on the histones? And a really great question, which we've been working on for some time, is, well, when you do have the phosphorylation on the methyltransferase, what signaling pathways is that phosphorylation coming from? Because we know how intricately that methylation is controlled along the genes. You can see it. I mean, it's gorgeous. You've got, with some of these things, you've got not much methylation at the start of the gene, but a whole stack of methylation at the end. How is that controlled? And it changes dynamically under the conditions that the cell finds itself in. So there must be phosphorylation, which is coming in from different parts of the signaling system to the methyltransferases that control that. And so we're very focused on now building out this complete network and system where the phosphorylation is coming to the methyltransferases and demethylases, which is acting on the actual histone methyltransferases that then act on the histones themselves, which then regulates the expression of genes.
Excellent, Mark. I think we from that we will leading to us to understand better entire system of the cell ultimately. So thank you again, Professor Marc Wilkins, to share with us today the sharing on the methylation and the functions, regulations, network, and I hope everyone have taken home the important message of how important in studying a particular, in this case, methylations, to understand how a cell work or the network connect to each other in the cell. So, and with that, we have come to the end of the seminar, and I also would like to invite all the participants, if you convenience, that you can switch on your camera so that we can take a photo with you.
Yeah, so I'd be delighted to see some of you and say hello.
And for your information, we have more than 130 participants today with us in this webinar.
Well, thanks everyone for coming along. It's been such a delight to meet you and be able to talk to you about some of our work.
We are great to have you, Mark, to share with us. So
so I also have my mask here, which I need to wear when I leave the office.
So you're in the office now, yeah? So some of participants, yeah, in the common area where you need to wear mask. So
yes.
Our technical team will take a photo and you inform us if it's done. Thank you.
Maybe we can all wave as the photo gets taken.
Okay, everyone ready? Okay.
Thank you when you're done.
Smile. 1, 2, 3. Okay. Okay, one more. Wait, yeah. Okay. One more. Okay, smile. 1, 2, 3. Okay, thank you.
Thank you, everyone, for joining today's conference. And on behalf of INBIOSIS and also Malaysian Proteomics Society, MAPS, I thank you again, and I hope you can join again in our future events. And today's webinar has been recorded, and you may be able to access it in the FP Live page and also INBIOSIS webpage. Thank you again, and everyone stay safe. Mark, please stay with us, yeah?
Sure. Thank you.
Thank you.